Hello, and welcome back to Introduction to Genetics and Evolution. So far, we've studied the inheritance of very simple genetic traits, traits whose variation is caused by maybe two alleles at a single locus, and we've looked at how to map that genetic variation. What I'd like to do now is get into some slightly more complicated facets. So let's look at how traits really are. Most traits are not, or most variation in most traits is not usually caused by variation at a single gene with two alleles. So we'll first talk about mutations as the source of all genetic variation, and then we'll go into what the genetic basis is of normal traits, and what are some complications rather than the overly simple model we used so far. So let's get started. If you go back somewhere between 3.5 and 4 billion years ago, we had what we believe to be the first origin of life. Now, we don't know exactly what that life looked like or exactly the conditions were, but we can imagine that maybe there was something along the lines of a single haploid genome. I say haploid, I mean having one copy of the genome, and a single primitive cell. Now, genome there was probably an exaggeration. It probably wasn't anything nearly as complicated as most of the genomes you see today, but it would have been some small piece. Now, the question I like to pose to you is, what would have to be true for you to define this thing as alive, this original cell as alive? How would you decide it was alive rather than inanimate? Well, the most likely answer people would say is it replicates, it reproduces, that it makes another copy of itself. So this one makes these two other daughter cells. And what it makes will be dictated by its gene. Now let's imagine hypothetically that the genetic code that was found in that original cell never ever changed over the 3.5 billion years of time. What would life look like on the planet today? Well, the answer is, if it never changed and if its genetic code is dictating what it can make, basically life would look like this today. There'd be a lot of copies of that simple haploid genome in cells all around the planet. That's obviously not what we see today. So there obviously has been introduction of some sort of new variation into the diversity we see on the planet today. Now, where does this variation come from? Well, this is the theme for today's lecture. Where does this variation come from? Well, what is the source of these changes in the genetic code? Well, the answer is mutations. Mutations are often defined in textbooks as a change to the DNA sequence, but very importantly, I'd like to emphasize they are the ultimate source of all genetic variation on the planet. Because again, if we have a single origin of life, which we do assume, a single origin of life, common ancestry of all existing life, then all the diversity we see has to have come from iterations of mutation from that original self-replicating form. Okay? So what is a mutation, or what is a mutant? I mean, you hear this word very often, both in common parlance and in the movies, things like that. Well, a mutant may be what you call your brother when he's being very annoying. Get off me, you mutant! I've probably heard that many times. Those of you who are fans of science fiction, a mutant in the comic world is an individual who possesses a genetic trait called an X gene that allows them to naturally develop superhuman powers and abilities. So there's a picture here from the comic X-Men. Many of you may have seen the movies from that. Probably not the definition we'll be using here in this class, as you'd guess. You may think of a mutant as someone who has an extreme disfiguration, such as extra fingers. So this individual right here has polydactyly. So if you count, they actually have 6 fingers. This is actually something that does come up. This is a mutation that does crop up periodically. It's a little harsh to refer to the person as a mutant because they have this, but it is a mutation which arises. Now, honestly, there's a little bit of truth to all those definitions, though less so for obviously the X-Men version. But again, the ultimate source of all variation is mutation. It is any sort of change in the genetic code. Okay? So you can think of it this way, that mutations happen, and they happen a lot. They happen far more frequently than you may guess, and you'll see in a subsequent video we'll talk about mutation rates. Now, one of the most common types that we'll focus on for this is an error in replication or in meiosis that leads to a change in a base, or a nucleotide. So imagine you have this stretch of sequence in a mom, okay? And let's say the mom is homozygous for this, so she has 2 copies of this stretch of sequence that starts with ATG. Now let's say in one of her eggs that makes the kid, there was this base change, went from A to G, such that this is actually what's in the kid. The kid then has a mutation. He or she has a nucleotide variant that was not found in either of the parents. So that is by definition a mutation. Now, importantly, let me emphasize what a mutation is not. So mutations do not happen when you need them. Right? They don't happen preferentially when you need them. You can imagine something like, oh, it would really be great to have 6 fingers. No, that doesn't happen at all. They are random with respect to need. Okay? Now, many mutations are bad. In fact, probably you could argue that most mutations even are bad, or at the very least, a large fraction. Now, why is that the case? If you think about it, our genetic code is not completely, but largely fairly streamlined. So if you introduce a random change, it's likely to be something that's bad. It could be something that doesn't really matter, but it's likely to be something that's bad. The analogy I often like to use is imagine that you have a car, right? And this car has come together over a very long period of time. And you take a hammer from way across the room and throw it towards the car. What are the odds that hammer will make the car better in some way? It's probably not 0, but it's extremely low. In contrast, what are the odds the hammer will either do nothing, or nothing substantive, or it will actually be bad for the car? Probably pretty high. So on average, we tend to assume that most mutations are either bad or they don't matter. Okay? I use one example here of one that doesn't matter. So many of you are familiar with the triplet code. So again, genes tend to code for proteins. They have a triplet code associated with it. And very often that third base in the code does not change which protein is produced. So if you have, as part of this code, AAA versus AAG, you would actually put the same amino acid into that protein. There actually would be no change in the final protein. So again, most mutations tend to fall into one of these two categories, either the bad or the don't matter. Now rarely, but it does happen, a mutation may be good, advantageous. Now, well, again, it doesn't arise when you need it, but it may just happen to be good. And it may not necessarily even be good at the time when it first arises, but it might end up being something that is useful in a subsequent generation. So thinking about that, let's imagine that a mutation has happened, and the new variant now exists in the population. So here's our first generation. Everybody has the same sequence. Okay. Now we have this one mutation, like the one I talked about previously, which is arisen in one egg cell. Now, as you'll notice, we now have the birth of a SNP, a single nucleotide polymorphism, because here all the sequences were exactly the same. Now we have one site which is polymorphic. It has 2 different variants there. Now let me ask you a question. Is it more important that this is transmitted meiotically or mitotically? Remember we talked about meiosis and mitosis way back in the beginning. Well, the answer is definitely meiotic, because if let's say, for example, this new mutation starts in this part of your hand, it spreads to the rest of your hand, it's never going to get passed on to the offspring. It's much more important it gets transmitted meiotically, so that it gets on to further generations and gets into generation 2, etc. So what happens then? What happens then is this new variant may be lethal. It may cause immediate death of the embryo. Right? That's a possibility. In the previous slide, I showed you one individual was heterozygous for it. Maybe that individual will die at the age of a couple of months in utero. It could cause some other sort of loss of fitness. It could be, yes, it doesn't immediately die, but they have extreme, gastrointestinal distress that makes it so they're not able to thrive as well. That's a possibility. These are definitely in the bad category, right? Now, it could be that this mutation causes no change in fitness, that this mutation is just the same as the parent in terms of overall probability of passing on the genes. So this would be what's referred to as the neutral category. And finally, maybe it causes superfertility. Maybe it makes it that this person is extremely attractive, or that when they do mate with an individual, they're much more likely to fertilize. This would definitely be the good category. And this would be much more like the X-Men version, presumably. So let's follow what happens after that. So again, we left off last time with generation 2. Here's this new mutation. Well, it may spread, it may get lost. It's possible that this individual would leave no offspring. Let's imagine this the case where I said this individual died at a very young age. Or it could just die for a random reason. Maybe this individual gets hit by a car at the age of 10. It could happen that this just never gets transmitted. That's one possibility. But let's assume it does get transmitted. Well, then, some individuals get it. This individual gets it here. This individual gets it here. In this case, we even start getting some homozygotes because this individual gets it and passes it on and has a kid with this one. So again, we have an individual here who's homozygous. We have a few others. This one seems to be spreading in this particular case. So it looks like maybe this particular mutation was advantageous or possibly neutral, but certainly not very bad. So this is the kind of thing that can happen with new mutations. So we have this new variation, it arises, it may just get immediately lost because it's bad. It may get immediately lost if it's neutral. It may even get immediately lost if it's good. Let's say, for example, this individual had super fertility, but again got hit by a car. That could happen. But for a lot of mutations, they will arise. You have the birth of a new SNP. They will be there in a population for some time. Now, over time, many rounds of this mutation, not just that individual one, will generate a lot of variation in natural population. And again, as I emphasized, a lot of mutations don't necessarily have an effect on fertility or lifespan, but they may still affect a phenotype. They may still affect how an organism looks, right? So let's say, for example, there was a mutation that slightly lengthened your earlobe. Maybe that wouldn't affect your probability of having kids whatsoever, passing on your genes, but it still could get passed on, and in that sense, it is still neutral, even though it has an effect. Now, I'm going to come back to this example in the next video, but I just want to introduce it. There's lots of genetic variation in things like, for example, human height. Now, some of that, of course, has to do with nutrition rather than genetics. But where does that variation come from? Is that actually all just from mutations at one gene? Well, take a look at the next video and see what you think. Thank you for joining.